So today we will discuss one of the important topic of CSIR NET syllabus, that is chromatin and in role of chromatin in gene expression. Some few questions of previous year question. So first question: phosphorylation of serine as well as the methylation and acetylation of lysine in histone tail affect the stability of the chromatin structure. So we know that the methylation leads to condensation of the chromosome or heterochromatinized formation, and acetylation leads to open up or euchromatin formation, right? So actually histones, we know that histones is a basic protein. Histone is a basic protein. Histone is a basic protein, and it is positively charged. It is positively charged, and due to this positively charged, histones interact with DNA. DNA is a negatively charged. DNA is negatively charged. That's why the interaction is possible. And histones actually reach in basic amino acid, that is arginine and lysine. And that's why the DNA histone interaction is possible. Now, see that here, the options, first option says is a methylated. First option, option 1. Option A, DNA is negatively charged. Option DNA is negatively charged. So this is correct. So DNA histone interaction is independent of net charge. No. DNA histones interaction dependent on charge interaction. So phosphorylation of serine increases DNA histone interaction. Now phosphorylation of serine is not correct. Now methylation and acetylation of lysine increases DNA histone interaction. Methylation leads to condensation and formation, and acetylation leads to open up of the chromatin. So it's not correct. Acetylation leads to chromatin formation. Now, so answer will be option A. Now next question. Question number 2. So 5-azacitidine. 5-aza, here see that question here, what the question says actually. Question actually saying that DNA methylation play important role in transcriptional regulation in vertebrates, and there is a inverse correlation in the DNA methylation, and we all know that DNA methylation is inversely with euchromatinized formation, because DNA methylation leads to heterochromatin formation. Now here is that the whereas there is a direct correlation between histone acetylation and increased transcription. Histone acetylation leads to increased transcription. Now here is that the uh, administration of 5-azacitidine to beta thalassemia patient. 5-azacitidine actually a it's a base analog. 5-azacitidine is a base analog of cytosine. 5-azacitidine is a base analog of cytosine. So 5-azacitidine 5-azacitidine is a base analog of cyto cytidine, base analog of cytidine. Cytidine is a nucleoside, right? And like same as 5-methyl thymine base analog, same. And instead of now here it can incorporate instead of cytidine. Another is here azacitidine. Azacitidine azacitidine cytidine used used in vitro used in vitro to remove methyl group from DNA. To remove methyl group from DNA, to remove ethyl group from DNA, and it also inhibit the DNA methyl transferase enzyme. DNA methyl transferase enzyme. So it's inhibit DNA methyl transferase enzyme. So these are three functions of 5-azacitidine. 5-azacitidine is a base analog of cytidine, and azacitidine is used for removing methyl group from the DNA strand, methyl group, and so it's, so see that option, which one of the not correct? Question is say which one is a not correct here. So not correct is first. See that here cell are exposed to 5-azacitidine, incorporates it into DNA in place of cytidine. So first option. Now second option is 5-azacitidine increase DNA methylation. Decrease DNA methylation. 5-azacitidine decrease DNA methylation. 5-azacitidine decrease DNA methylation. 5-azacitidine promote histone acetylation. Yes, 5-azacitidine promote histone acetylation. 5-azacitidine decreases DNA methylation, and cells exposed to 5-azacitidine incorporates into DNA in place of cytidine because it acts as a base analog. And 5-azacitidine, so option 4 is 5-azacitidine does not promote gene expression. So it's not correct. So answer will be option 4 is the correct answer. Now question, next question. So genes are translocated to heterochromatinized region. Generally heterochromatins are gene generally silenced, and there is a high chance of methylation is there. And we know that two type of enzymes is here there. So see that first question. So interest was translocated into the centromeric region. Centromeric region is a heterochromatinized general region, and it is silenced. So mutagenesis leading to loss of function of the following target gene. Loss of function of the following target gene and of interest and new locus. The mutation in histone deacetylase. Mutation in the mutation expression of the gene of interest. And now see the option A, mutation in histone deacetylase, mutation in histone acetyl transferase, mutation in histone H3 lysine methylation, loss of dicer and RNA processing enzyme. Now the question is say gene expression, in which case gene will be, above cases gene will be expressed. Now option C. First of all, look at the option C. So you can see that here H3K lysine in the K9 methylation leads to chromatin condensation, right? So generally, if it is a mutation is here, so gene expression will be possible. So mutation in histone no, H3 lysine, there is a gene mutation is possible. So this will be correct. Mutation in histone deacetylase. Yes, there is two type of enzymes is there. Had histone acetyl transferase, histone acetyl transferase, which transfer acetyl group and increase the gene expression level, and the HDAC, histone deacetylase, histone deacetylase, tylase, remove acetyl group, remove acetyl group, tyl group leads to gene silencing, right? So the option B, see that option B says that mutation in histone acetyl transferase. If it is a mutation in histone acetyl transferase, so it's a gene expression could allow the gene expression. So mutation in histone deacetyl transferase not lead, it's not leads to gene expression. So option B is wrong. So B is wrong. B is wrong. Now see that A. Now here is that it's a Saccharomyces pombe. S pombe, S pombe, POMB, S pombe, two type of pombe. The HDACs are histone deacetylase. HDACs are encoded by coded coded by HDAC, HDA, HDA1, CLR, CLR3, and CLR6. CLR6. CLR, what's the stand for full form of CLR? CLR stands for here. CLR equal to cryptic loci regulator. Cryptic loci regulator. So these two, the CLR3 is a HDAC, histone deacetylase. So if it is a mutation, histone deacetylase enzyme, histone deacetylase enzyme. So it leads to gene expression, histone deacetylase. It's remove acetyl group, right? So answer will be option B is the correct answer. Option B is the correct answer. So option B. Now next question. Now see that the following statement are made about chromatin remodeling in human cell. It's a local chromatin conformation may play more important role than local DNA sequence of the promoter. Option B, histone in nucleosome can undergo many different covalent modification in turn alter the chromatin architecture locally. Now, chromatin remodeling is a developmentally regulated passive process which does not require ATP. So, first of all, see that the chromatin remodeling actually needed to open up the DNA, right? DNA, if different methods are there for chromatin remodeling, like ejection, repositioning, ejection, unwrapping, dimer exchange, and dimer ejection. These are different method where the DNA actually stand open up for for accessibility of the genome, and it's different remodeler families are there. So among them are SNF family, ISWI family, CHD family, INO80 family. These four are the remodeler families. So remodeler families always requires an ATPase domain is there for all remodeler family content, an ATPase domain. This ATPase domain is absolutely necessary for their function. So local chromatin conformation may play. So first of all, that this option C, chromatin remodeling is developmental. It's a passive process. No, it's not a passive process. All required an ATP dependent ATPase domain is there. This ATPase domain actually involved for their remodeling function. So remodeler is ATP dependent process. So option C, if we remove from here, so answer is option 3, that is ABD. But see that a local chromatin conformation may play important role than the local DNA. Yes, histones and nucleosome can many different modifications are there. This modification leads to different types like acetylation, methylation. Different modifications are there. Several histone variants are exist. So answer will be A, B, and D. Now, next is, so there is a remodeler. So see that how chromatin remodeling occurs. So chromatin remodeler. So these are deposition. Now these are remodeler. Remodeler needed ATP to ADP converting. And now here see that DNA binding protein. These are DBP. Now these are, see that first one, repositioning. Repositioning is Now here this now DNA, now this DNA is now is going to here. See that this here open up and then it's accessible for the DNA binding protein, right? DBP, and then ejection, repositioning. This is site exposure. Now altered composition. Here see that histone variance is added. So all are leads to the open up of the chromatin, so accessibility of the genome, right? So another important is the all the remodelers have some different uh, all remodeler contain 5 basic properties. All remodeler, all chromatin remodeler remodeler have 4 basic properties. One is that an affinity for, they must have an affinity for nucleosome, affinity for nucleosome. Number 2, domain, they must contain a domain that recognize covalent histone, recognize covalent histone modification, histone modification covalent histone modification. Number 3, similar DNA dependent ATPase domain. They must have a DNA dependent ATPase domain. ATPase domain. They must have a DNA dependent ATPase domain and domain or protein or protein domain or protein for interaction with other chromatin, for interaction with other chromatin or transcription factor. These are the 4 basic properties must be present in a one remodeler, right? So remodelers are the family. So remodelers have 4 group of family: SWI SNF family, ISWI family, CHD family, and INO 80 family group of remodelers, right? Next is